Okay, I'm going to do some short videos on how to do your student experiment write-up because I've got an awful lot of common issues that I'm addressing with different people. So first of all, we're going to do rationale. So for rationale, you have to show me your understanding of the issue and talk to me about how you've designed an experiment to test something about the issue. Now, the issue that we had was fermentation. Remember here on our notes? We actually went in and we learnt about respiration and fermentation. We learnt about the fermentation equation. This is it here, anaerobic respiration. We actually want this one down here, which is alcoholic fermentation. You learnt how that actual reaction happens. You learnt that fermentation, sorry about that, this is a quick video, is respiration without oxygen. And that is the sort of information that you actually need to base your rationale on. So you need to show and explain to the person who's reading what fermentation is. What sort of reaction is it? Why does it need sugar? Why do we even need to have fermentation at all? Remember, it's got to do with recharging ATP. What are the bubbles that you're actually counting? Why is that important? Why are we counting bubbles? How does that relate to our fermentation equation? Think about what our substrates are. That's what we have to begin with, and our products. You have to actually show some understanding about that. Okay, why are we counting bubbles? What am I actually counting bubbles for? Look at your alcoholic fermentation equation here. What is it producing as it breaks up glucose or other types of sugars? You need to actually talk about that and show your understanding. So the rationale needs to show that you can explain to the reader what is fermentation, how does that relate to what we're actually doing in our experiment. I hope that makes sense. So make sure you do that. You have plenty of videos that you had on those notes and on your homework sheets that you can watch to re-explain fermentation, like this one here by the Amoeba Sisters. That is this very first one here that you had to watch. So go back, look at your notes, and actually get in there and explain fermentation to me. So that's the first part of your rationale. Then you have to tell me what you actually changed to investigate something that's affecting fermentation. It was either its exposure to a substance that might be toxic. It was, just trying to actually think here because I'm having a brain dead day. Different types of sugar or different concentrations of sugar. So you're affecting the fermentation reaction rate by changing one of those independent variables. So you have to talk to me about what you changed, how you changed it, and what else you controlled in order to make sure it was a fair test while you did it. So anything else that you know would affect fermentation needs to be controlled, and you need to talk about that in your rationale. Okay, think, why did we measure everything so carefully? Why did we keep temperature controlled really well? Go into that in detail for your rationale. Background theory is fermentation. Last but not least, you had to stick your neck out and make an educated guess. You had to tell me, if I change this in this way, then this will happen. I think this will happen because, and you have to back it up with basic research. So for all of the things that you had to research, if you look in your OneNote, just going to go over here. For toxic copper, I gave you information about that. And remember, it's used as a fungicide and stuff, so it's often found on fruits that are used then to make wine. For different types of sugar, there's that, plus I've also added in some extra pictures. And for concentrations, here's a picture of what happens if you actually have too much sugar. Here's a picture of what may happen to cells if you have them in a solution that has either too much sugar or too much salt in it. It actually ends up with the water being sucked out of the cell. Well, it's not very productive if the cell is being sucked dry because it's got too much dissolved sugar around it. So they were the things that you were actually investigating. You've already been given the basic theory to this, guys. You just had to actually talk about it in your rationale. So all of this is in here, has been since the beginning. I want you to go through it and read up on it. So that is essentially what you need to do for your rationale. The next thing I'm going to be also added in on there, I'll just put it in here, you have to detail for me how you modified it. So whatever you're investigating, you need to explain to me why you modified it that way and how you made sure when you, say, changed the different types of sugars or added in copper or whatever, how you made sure it was a fair test when you modified my method, how you made sure you maintained it being a fair test. Add that in, and my next video is going to be on processing data and making tables.